thing on. Can see my chat. We're recording. Okay, I think I'm set up good. Okay, so chapter 9 is about proteins. We're now really getting into biochemistry here with proteins in the body. The human body has about 100,000 different types of proteins. That's an amazing amount. A cell has about 9,000 proteins in it. It's about 15% of the cell mass. And if you dried it out, most of our bodies are liquid, right? If you dried it out, the proteins are actually 50% of the dry cell mass. Proteins have carbons and hydrogens, oxygens, nitrogens, and some even have sulfur in them. We'll see more about this in a minute. Proteins are used for the synthesis of enzymes, the building up of hormones, components of blood, creating components of blood, maintenance and repair in the body, as well as creation of new tissue, and for energy as well. So a protein is defined as a natural unbranched polymer made up of amino acids. So each one of these different colored balls is an amino acid, and it's a polymer. They're all chained together like this. And here you can see these are individual amino acids. You've got the carbonyl group right here, and you got the amino group on that side with an R chain hanging off. And they're all connected to each other with what's called a peptide bond. But a peptide bond, as you can see, is nothing more than just an amide bond. So you've got the amino group here and the carbonyl group here. That is an amide linkage. But in proteins, they call them peptide bonds. And the same here, you got an amino group here and a carbonyl group here. This is a amino or amide linkage as well. As usual, just pipe up if you have any questions or comments or anything. So amino acids, the building blocks of these proteins, as I think we've already talked about amino acids, they have this amine group here on one end, and they have this carboxyl group on the other side. Most of the amino acids in proteins are alpha amino acids. Alpha amino acids are when both of these functional groups are attached to the same carbon. And then you've got some side chain hanging off of here as well. Notice that that makes this carbon a chiral center. It's got some R chain, an amino group, a carboxyl group, and a hydrogen. So this is a chiral carbon in that particular case. Most, but not all. There's about 700 naturally occurring amino acids. There's 20 major ones that are present in proteins that make up proteins. So those are the 20 that we're going to focus on. There are different types of amino acids. There are non-polar. The side chain is not polar. There are polar amino acids where the side chain is polar, but it's electrically neutral, so it still has a net charge of 0. A polar acidic amino acid, the side chain has another carboxyl group on it, and a polar basic amino acid, the side chain has another amino group on it. So here are the nonpolar amino acids. And these usually get referred to by their common names. I'm not going to make you guys memorize these or anything like that. But just to be familiar with them, if I ask you a question on the test, I will give you the structure. So I'm not going to expect you to memorize what valine looks like or whatever. But these are the nonpolar amino acids. We've got glycine. this gets abbreviated GLY. Alanine is not Alabama, it's ALA. Valine is Val, leucine is Lou, isoleucine is ILE, so forth and so on here. And you can see these R chains, which are in red, are quite different from the other ones. This one actually, proline is a ring. Phenylaniline has a phenyl group off of it. Methionine, I guess they say pronounce it, has a sulfur in it, which you would expect with the word meth in it. And tryptophan, you've got a fused ring on that R chain. So these R chains definitely can be quite different, but notice that all of these R chains are nonpolar. Polar amino acids, you can see here, this is serine, or I guess serine. There's an OH group here, so there is going to be a partial negative charge on that more stronger electronegativity of that oxygen. Here with the sulfur and cysteine. Here we have another OH group here. Here, glutamine has a carbonyl group and an amino group on the end. So this will be polar as well. And same with asparagine. And you can see the 3-letter designations in here. Polar acidic amino acids, like I said, they have a carboxyl group on the side chain. So aspartic acid is one of these. So you've got a carboxyl group here, you've got an amino group here, but in this R chain, there's also a carboxyl group. And so this is a polar acidic acid, amino acid. And the same for the glutamic acid. It's got this group here as well. The only difference here is an extra carbon in the chain. And then polar basic ones have an amino group in them. In the case of histidine, you've got an amine, two nitrogens in this ring, and here's your amino group with the hydrogen on it. Lysine is a straight chain with an amino group on the end. And then arginine has 2 here. We've got an NH double bonded to the carbon, and then an amino group bonded there as well. And then here's just all of them listed again with their 3-letter abbreviations. Okay, so chirality, like I already mentioned, this carbon here is going to be chiral 19 out of 20 times. Glycine is the one that is not. It's got 2 hydrogens. And so this is not a chiral carbon, but all 19 others of them have a different R chain from an amino group, carboxyl group, and a hydrogen. So this is a chiral atom carbon. When you draw Fisher projections of amino acids, you put the carboxyl group at the top. And then if your amino group ends up on the left, it's an L. And if it's on the right, it is a D. So this is L-alanine, and this is D-alanine. Any questions there? But amino acids do have acid and base properties. You've got a basic group here in the amine group, and you have an acidic group here with the carboxyl group. And the amino group can accept a proton from this carboxyl group. And so you can end up with what is called a Zwitter ion. Zwitter is the German word for double. And so in a neutral pH solution, you can end up with this having the form of NH3 plus instead of NH2 and COO minus. And this is called an internal acid-base reaction because it's actually happening within the same molecule. So this proton is being donated over to here. Now, keep in mind that this is actually like a resonance structure where it's going to be flipping back and forth. This is just one state. The other state would be back to this way. You know, it's our way of rendering it. If you're in a low pH solution, you're going to have an excess of hydrogen protons. And so a hydrogen will attach to this negative thing. So you'll end up with an NH3 plus over here, but also a hydrogen attached to this COO minus group on this side. And if you're in a high pH situation, you'll end up losing the proton over here, but it doesn't pick up the proton again. So you end up with an NH2, but then a COO minus on this side. Any questions there at all? So here's a practice question for you. This is alanine. If it was in a pH 11 solution, what would it look like? So let's try to draw that out for me and let me know when you're done. Oh, it's cool, Danielle. You're doing a few this year. Which class did you sign up for this year? If you remember. Oh, there's a yoga class? I didn't notice that. That's cool. Yeah, I sent them a whole list. I'm like, I could talk about chemistry, I could talk about computer science, I could talk about Judaism. I don't remember what all. They were like, all of them look great. So I just picked one. All right, what are you guys about done? Did you say it would look like this? So this is a basic pH. And so you're going to end up with not having a extra proton over here, nor will you have a proton over on the carboxyl group either. Okay, that was in the case of a neutral atom. In the case of an acidic polar amino acid, you end up with 4 different forms. And so here is our Zwitter ion. And you see here, this is an example of an acidic polar amino acid because we've got this carboxyl group here. This still does the usual thing. This is our Zwitter ion formation. In a low pH, you're going to add, you're going to add a hydrogen onto this COO minus side because you have excess hydronium ions. In a slightly higher pH, above neutral, you're going to have the hydronium ion on this carboxyl group fall off. So you're going to end up with Cu minus Cu minus. Call this Cu. And then when you get to even higher pH, you're going to lose that extra hydronium ion off of that amino group, and you're going to have minus and a minus, but then NH2. So there's 4 different forms here depending on the pH. Any questions there? And you have a similar thing with basic polar amino acids. There are 4 different forms. So here is our Zwitter ion with a net charge of 0. And this is the case. Remember, a basic polar amino acid has an amino group in the side chain. And so there's actually 2 forms to this Zwitter ion. You can have the minus on the carboxyl group and the plus on the side chain amino group, or it could be the side chain over here as well. So there's actually 2 different forms to this. But then if you lower the pH, you're going to get the hydronium ion adding over here to the amino group. And if you lower the pH even more, then you're going to get a hydronium ion adding to your COO minus group as well. And then if you drive the pH up really high, you're going to lose the hydrogen, the extra hydrogen off of both of these amino groups. Cool. So the isoelectric point is the pH where 99% of the ions are in the Zwitter ion form. You'll never get quite 100%, but it's the pH where 99% of them will be there. And so depending on the pH, you'll have these ions at different states, as we just looked at. And there's actually a process called electrophoresis, which will separate your molecules from each other. And you'll have the positive species will move to the anode and the negative species will move to the cathode. So in this example, we have a mixture of lysine, phenylene, and glucosine, and they're mixed together. But at a pH of 5.5, you're going to have the lysine will move towards the anode, the negative electrode, because it has a positive charge on both of these amino groups. And the glucine is going to move to, or glutamine will move towards the positive side because it's at this pH, it's going to have both of their carboxyl groups are missing their hydrogens. And at this pH, phenylaniline is neutral. There's a plus on this side, a minus on this side. So that balances itself out. And so it doesn't go anywhere. So you can actually separate these at a particular pH because you've driven these to different forms. And this is called electrophoresis. Any questions there at all? All right. There is a unique amino acid. This amino acid, I think they said to call it cysteine. So cysteine can dimerize, or 2 of them come together and bond to form what's called cysteine. And cysteine, they bond across these sulfur, these are sulfuric alcohol groups. and you end up with a disulfide bond. So these kind of flip on their sides, if you will, and that SH and that SH come together, lose their hydrogens, and it forms this disulfide bond, and this forms a amino acid that's called, it's a dimer amino acid called cysteine. And we will see that this is biologically significant in a few minutes. So this is a different than the others because it so readily dimerizes like this. Okay, peptides, like we said, are just amide linkages with one another. We've got the carboxyl group on one molecule and an amino group on the other, and they will merge together. And this is a dehydrolysis because you're getting water off and you form this amide bond right here. Peptides are two or more amino acids chained together via those peptide or amide bonds. And so you just have your amino acids in that chain. These were the different colored balls in that first picture we looked at. And then you've got your amide linkage or peptide bond in between each one. So a dipeptide would be two amino acids together. Tri is three. A oligopeptide is 10 to 20 amino acids chained together. And then larger than that, we just call them polypeptides. Those are long chains of amino acids. Most proteins are polypeptides because they're huge. So the way these get rendered, I'm trying to remember what I'm doing with this slide here, is that you put the amino group on one end of the chain and your carboxyl group on the other. Usually this is put on the right and this is on the left. And this is called the N-terminal end for the amino group. And this is called the C-terminal end for this carbon and this carboxyl group. And this is an example of glycine, alanine, and serine together. So you see here we've got this peptide bond, an amide linkage between glycine and alanine. And then we have a peptide bond between alanine and serine. See that? And you've got your N terminal down here and your C terminal on the other end. This also becomes important. And this gets rendered with just the abbreviations. So this is called a gly-ala-ser peptide. This would be a tripeptide. Any questions there at all? So this is called the peptide backbone. And then you just have all of your R groups hanging off of that backbone, whatever they happen to be. So if I give you glycine, alanine, and valine, can you draw what this tripeptide would look like? I'll give you a few minutes to try your hand at that. Just put in the chat that you're done when you're ready. How you guys doing? You guys need a few more minutes? You're a quiet bunch tonight. Okay, Lydia, Danielle, baby ready? Everybody else, how you doing? Ready to move on? So you should have something that looks like this. So we have this alanine here is going to have this amide linkage to the glycine here. That's this glycine is just a CH on either side because its amino group is connected in to the alanine. And then valine over here will be connected into the glycine in here with the linkage. That close to what you guys got? Okay. Constitutional isomers or structural isomers can exist with these chains, and they do have different chemical and physical properties because of it. So an alanine glycine is not the same as a glycine alanine, and they will have different chemical and physical properties. The number of possible isomers is equal to n factorial, where n is the number of unique kinds of amino acids in the chain. The emphasis here is on unique. So let's say you had five different amino acids chained together, then 5 factorial is 5 times 4 times 3 times 2 times 1, which is 120. So you could have 120 different combinations of this. I probably should have put an example in, but if you had an alanine glycine alanine again, that would have the same number of isomers as just an alanine glycine because you don't count the duplicate ones. So it's unique kinds of amino acids. Does that make sense? Okay, naming rules. The C terminal keeps its full amino acid name. All the other amino acids have names that end in YL. and you begin the naming sequence at the left N terminal. So alanine glycine becomes alanyl glycine, and glycine aniline or alanine becomes glycyl aniline. See how it's flipped because the sides are flipped? And then if you had a tripeptide of valine, serine, and aspartic acid, then it would be valyl seryl aspartic acid. Fairly straightforward. Okay, we said that these peptides, these proteins, can form hormones. Here's an example of two hormones. Oxytocin regulates uterine contractions and also lactation. And vasopressin regulates the elimination of water through the kidneys, and therefore it affects blood pressure. Both of these peptide hormones, or protein hormones, are generated in the pituitary gland, which is in the brain. You can see it right here in this example. But notice that they're almost identical. So the difference here is in the third and the eighth amino acid. In the third amino acid, oxytocin has isoleucine in it, but vasopressin has phenylalanine in it. And the eighth amino acid is different. Oxytocin has leucine, and vasopressin has arginine. But other than that, they're identical in their structure. And oh, by the way, here's this disulfide bond between these two cysteine guys. So there's that too. We'll talk about that more in a minute. But so just a little bit of a chemical change, and they're used, these hormones are used two very different ways. They could also be used as neurotransmitters. Enkephalins are produced in the brain, and they bind at receptor sites in the brain in order to reduce pain. There is a metenkephalin, which has this, what's this, 1, 2, 3, 4, 5. So this is so this would be an oligopeptide. And this has tyrosine, glycine, phenylalanine, and methionine is the structure of this one. This is called and it's, this methionine is where it gets the met from. A leuenkephalin is tyrosine, glycine, phenylalanine, and leucine. Both of these chemicals produced in the brain play a role in a runner's high. So we were talking about running a few weeks ago, how your body produces hormones in your brain when you run, and that's where some people get that runner's high. These chemicals, when you're running and you kind of get that high, will reduce pain. They'll activate and bind to these pain receptor sites and reduce pain while you're running. Morphine and codeine, which we've looked at before, both bind to these same sites and therefore have very strong pain reduction properties and are very addictive because of how strong they are. They can also act as antioxidants. So glutathione is made up of a glucine, a cysteine, and a glycine. This exists in most cells in the body, and it regulates the oxidation reduction reactions going on in the body. These antioxidants protect cell contents from peroxides and superoxides. And what happens is that they get oxidized instead of the other species in your cell. So remember we talked about antioxidants. Sometimes there's natural antioxidants in foods, and there's also synthetic ones that get added to foods to keep them from oxidizing. Those synthetic ones oxidize instead of the food material. This is the same kind of concept. These get oxidized rather than the other species inside the cell, and therefore it protects them. So glucine is bonded to the cysteine via side chain carbonyl group rather than the alpha carbon. So this is one thing that's a little different, is it's not the carboxyl group or the amino group tying in here. This is actually the side chain. This right here is the side chain attaching to the cysteine. Okay, so proteins are usually 50 or more amino acids. Large proteins will have like 400 to 500 in a chain. Smaller proteins will be around maybe 50 to 100 amino acids linked together. A monomeric protein only has one peptide chain. Multimeric proteins have 2 or more peptide chains. So here's an example of insulin. This is a multimeric protein because we've got 2 chains here. And you see here that the first chain has got 21. This A chain here has got 21 amino acids. And the B chain has 30 amino acids. And notice that you've got a disulfide bond between these 2 cysteines and these 2 cysteines. And then there's also one within the same chain like this. Oops, I keep accidentally clicking the button. So this is insulin. This is the chemical structure of insulin. Any questions there? I forgot to put my slide numbers in here. Simple versus conjugated proteins. Simple proteins only have amino acids. So that's like this. These are all amino acids. Conjugated proteins contain some non-amino acids in the chain. These non-amino acids are called prosthetic groups. Think about it as like, a prosthetic is like a fake arm or fake leg or something. They're not, part of your body per se. That's kind of the idea behind it. There are lipoproteins that have lipids in them. There are glycoproteins that have carbs. And there are metalloproteins that have metals. These are the 3 groups of conjugated proteins. And you can see over here, I threw in a picture here of the lipoproteins. You can see here the blue is the protein chain, but there's cholesterol esters, there's cholesterol itself, there's triglycerides and phospholipids all together making up this lipoprotein. 2 examples of lipoprotein that we've talked about before is LDL and HDL cholesterol. So LDL is the lipoprotein that brings cholesterol out into your body when it's needed, and HDL is when it pulls it back in from out in your body. And this is why LDL, if it gets too high, is bad, because you're depositing a lot of cholesterol into your system. Glycoprotein, here's an example. We've got this monosaccharide attached onto the chain here. And an example of a metalloprotein is hemoglobin. So here we've seen this guy before. We've got this very complicated structure, and there's an iron that sets in here in the middle, and this is where the oxygen attaches for the hemoglobin to carry that oxygen throughout the body. That is a protein, it's a metalloprotein that carries the oxygen through the body. Any questions there at all? Okay, so proteins have primary, secondary, tertiary, and sometimes quaternary structures. The primary structure of a protein is just the order of the amino acid linkages in the protein. So you can see here again these different colored balls just indicating the different amino acids. So this is the primary structure. So we go back to here, we can see here these different ones in here. This is the primary structure, is just which amino acids are in what sequence. Each protein has its own unique sequence of amino acids, and the protein is defined by how many amino acids it has, the different types of amino acids that it has, and the order in which they're combined. The primary structure is the same in an organism no matter where they are generated in the body. And they are often the same across species as well. So insulin, for example, is very similar in cows, pigs, sheep, horses, and humans. But if your body produces insulin in two different places, they're both going to be the same no matter where they're generated at. And the body will always produce them in the same structure every time. So insulin has 51 amino acids. This is 1.55 times 10 to the 66 power possible sequences of those 51 amino acids. That's a huge number, but the body always builds insulin exactly the same way. And I think it's next week we will get into how that actually happens. And I think it's pretty amazing. Any questions there about primary structure? Secondary structure then happens when there's interactions within elements on the same chain. There's two possible ones in proteins. There's an alpha helix and there's a beta pleated sheet. An alpha helix is a coil shape, just like you see over here, and it's maintained by hydrogen bonds between the amino and carboxyl groups on the various amino acids. So you see here there's an amino group that has an attraction to this carbonyl group. Here's an amino group that has an attraction to this carbonyl group, so forth and so on. And this causes that chain to curl into a helix formation. But they can also be folded in on itself like this. And that's what's called a beta pleated sheet. And this is caused by hydrogen bonds between these different amino acids. And I had to throw in there like a kilt, because you pleat up a kilt when you put it on. Any questions there? You guys still there? Did I put you to sleep?
I'm here.
We're all good?
Still here.
All right. Most proteins are a mix of these. So you can have a helix here, and then some random structure, and then another helix, some more random structure, then a belted pleat, and then another random structure out here as well. So most proteins are a mix of both of those secondary structures like this. And these lines are just showing you where those attractions are at. Those lines are part of the actual molecule. Those are the attractions between them. And then we can have tertiary structure. So it's the overall 3D shape. And the book gave an analogy here, which I thought was really good, with a phone cord. The primary structure is just the straight phone cord line. The secondary is the fact that it's curled up like this. And then the tertiary structure is how this kind of wraps around on itself. The only problem with this metaphor is that, as you know, the next generation may not even know what these are anymore. But you guys know what a phone cord looks like, right? So I think it's a pretty good analogy. The tertiary structure is the overall 3D shape from interactions between these side chains and the amino acid. And what can affect tertiary structure is covalent disulfide bonds that we saw with the cysteine, electrostatic attractions, also called salt bridges, hydrogen bonding, and then hydrophobic attractions. Do I have a slide for all of this? Okay, I do. Good. So we'll look at those right now. If I can keep my thing from going crazy. So the disulfide bonds we already talked about. That's where we get this bridging, this between two cysteine amino acids. They could be within the same chain and they can occur across chains, as we saw in a previous example. These are the strongest of the tertiary interactions because these are actual physical chemical bonds, not just attractions. So this side gets close enough to this side that they merge and form this disulfide bridge between the two sides. Electrostatic attractions between them. We've got a chain down here. This is lysine, and this is I'm trying to remember what ASP is. Is aspartame? And we have this carboxyl group on one side, on its side chain, and we've got an amino group on this lysine side. So these two will be attract each other because one has got a partial negative charge and this has got a positive charge on it. So this will cause those two chains to kind of form together in 3D space. And then we can have hydrogen bonding as well. So here we have serine has a hydroxy group, and this oxygen is forming a hydrogen bond, or I'm sorry, this hydrogen is forming a hydrogen bond with the oxygen on this carbonyl group here on the other chain. And so hydrogen bonding contributes to this 3D structure as well. And then finally, hydrophobic interactions. So when you have two nonpolar side chains, they can clump together inside of the chain in order to stay away from any water. So they will want to be away from water. They're hydrophobic. And so this will cause the chains to kind of, wrap around and cluster together to sort of keep away from the water. And an example of this in the real world is when you fry an egg. They have this white with a clear exterior. And this is because the hydrophobic proteins will clump together to avoid the aqueous egg. So you see this white on the outside are these chains clumping together, trying to stay away from this aqueous, the clear side of things. This is what actually causes this to happen when you're cooking an egg. Isn't that kind of cool? So here's all of the interactions together. So we have a salt bridge or an ionic bond across these two, so that causes this chain to kind of shape like this. Here we have a hydrophobic center. These two nonpolar guys want to stay together and stay away from all the water that's out here. And so it causes it to do a shape like that. Here we have a disulfide bridge, which is going to pull these two sides of the chain together. And here we have hydrogen bonding, which is causing this to kind of loop or bend around like that. So you get this sort of shape in 3D, 3 dimensions, based on these different interactions. Any questions there? Does all make sense? You can have a quaternary structure, and this is when you have proteins with multiple chains in them. And these will attract with each other, and you'll get another additional layer of complexity as they interact with each other. So you can see here there's like a light blue chain, and then you've got a darker blue chain, and these will form this quaternary structure. It's kind of like tangling two phone cords together. That's a good analogy. Never used that before. That's kind of what's happening there. If your phone cord kind of had two, then they would form this quaternary structure. So again, just to reiterate, we have the primary structure, which is just a sequence of amino acids. We have the secondary structure, which could be an alpha helix or a beta pleated sheet. Tertiary structure as these guys interact with each other. And then you could have a quaternary if you do have multiple chains, and a multimeric protein is a protein with multiple chains, then you can get a quaternary structure out of that as well. Any questions there at all? There are two main forms of proteins. You have fibrous proteins and globular proteins. Fibrous proteins are long strands or sheets, so they tend to look like this, and they've got a more linear structure to them. Globular proteins are spherical or kind of look like globs. They look more like this. So as you can imagine, hemoglobin, which do you think hemoglobin looks more like? Is hemoglobin a fibrous protein or a globular protein?
A globular?
Yeah, it's more of a globular protein. Fibrous proteins build structure in the body, and globular proteins usually are used in storage and transport in the body. I think I got a slide for that. Fibrous proteins tend to be water insoluble. Globular are water soluble. This allows them to move through your bloodstream and other fluids in your body. Fibrous proteins usually have a single secondary structure. Globulars will have several. Fibrous proteins provide structure. Globular proteins are used for metabolism, catalysis, transport, and regulation of functions in the body. There are far more globular proteins than fibrous proteins, but most proteins in the body are of the type fibrous, because you've got muscles, ligaments, your skin is made out of fibrous proteins. There's a lot of structure in our bodies, and so that's why you've got so much fibrous proteins in your body. Fibrous proteins. Alpha keratin is its shape is like coiled microfilaments. You can see here all these different colored strands of these different proteins all shunted together. Alpha keratin is used in the formation of hair, feathers, wool, nails, claws, scales, horns, shells, quills, and hooves, as some examples. So all of these kind of get that structure from alpha keratin. How many disulfide bonds across these different chains in here determines how hard it is. So something like a horn, it's harder than hair or wool. It's going to have more disulfide bonds, making this much more rigid than hair or wool. Collagen makes up our tendons, ligaments, blood vessels, and our skin. It has a shape of a triple helix of 3 chains kind of wound together. Collagen makes up about 30% of the proteins in our body. As you can imagine, most of our body is tendons, ligaments, blood vessels, and skin. The cross-linking gives strength to these fibrous proteins. And those cross-linking increases with age. So this is why skin gets less supple as we age. Spoiler alert, guys, when you get older, your skin will not be quite as supple as it is right now. And this is also what happens in the tanning process. When you take a hide, which is much more flexible, and you tan it, it becomes the rigid leather. And that's because in that tanning process, you create more cross linkages between these chains, making it a harder substance. Globular proteins. So hemoglobin is a globular one, like we already said. Here's another picture of a heme. This is used to transport oxygen from the lungs to the tissue in the body. That oxygen comes in and sets right on that iron center and is carried throughout the bloodstream. Myoglobin is another globular protein that lives in our muscles, and it has a greater affinity for oxygen than hemoglobin. So when the blood comes through, the oxygen will transfer from that iron over to the myoglobin. And this is how oxygen is stored in your muscles to be used as you expend energy. Chemical reactions. So hydrolysis will reverse the peptide bonds and form free amino acids. This happens during digestion so that the amino acids can be absorbed into the intestine walls. And your intestine can't absorb these large proteins. It has to be broken down first into the individual amino acids, and then those amino acids get sent to your liver as building blocks to later make more protein. So you see here an example. We have this tripeptide with the application of water that's in your body, your stomach acid, so you get an acidic environment, and your body has heat as well, your stomach. This then breaks it apart into these individual amino acids, and these can absorb into your bloodstream through your intestinal walls and be sent to the liver for processing. So that's why I think I said a few weeks ago that our bodies are like a puzzle. It's like we take in the puzzle, it gets broken apart, and then it gets rebuilt again. That's basically all you're doing when you ingest proteins, is it just breaks them down and builds new proteins as the body needs them. Denaturation is the process where a protein loses its structure and it goes from having this nice organized structure into just a random protein strand. Denaturation, you lose the secondary, tertiary, and quaternary structures in those proteins. This affects the biochemical functionality because you can see that it's just not in the same. So it has the same primary structure still. We haven't broken the amino acids out, but it's lost all of these attractions in here that gives it its particular shape, which does affect its biochemical functionality. If you denature a protein, it becomes less water soluble. This is what happens when you cook proteins. You're making it easier to digest because it's not in these rigid structures anymore. It's been broken out into this sort of just random strand. And this is why if you run a fever consistently greater than 106 Fahrenheit for an extended period of time, it will denature the enzymes in your body, which are proteins. And if it goes on for long enough, it can kill you because your body basically can't function anymore. You've destroyed all of your biochemistry in your body. This is why high fevers kill you, is your body just isn't able to do the chemistry it needs to do anymore. And this is again an example when you're cooking this egg, you're denaturing the proteins and you end up getting these just random strands building out here. UV light also, UV light denatures skin, and this is what causes sunburns. Denatured proteins and yogurt gives it yogurt its semi-solid consistency. Acids and bases in your eye, if you get acids or bases in your eye, this denatures the proteins in your eye, and this can cause clouded corneas and ultimately blindness. Heat and or ultraviolet radiation also denatures bacterial proteins, which kills them. So this is why oftentimes you heat up things or you expose them to UV light in order to kill any bacteria that is there. Alcohol is an excellent agent for denaturing bacteria, and this is why it's used as a topical disinfectant. Whenever you get a shot or anything, is it will reduce the chance of a bacterial infection. Glycoproteins. So we said glycoproteins are conjugated proteins with carbohydrates or carbohydrate derivatives on them. Example of this is collagen and also the immunoglobulins in our system that are used to fight infection are glycoproteins. Collagen, remember collagen we just talked about has this fiber structure to it. The carbs in these protein chains cause the cross-linking between those triple helices that we talked about, and this forms that more rigid structure in there. If you boil collagen in water under basic conditions, you will produce gelatin. And what happens there is the heat is denaturing that triple helix structure. Meats become more tender because it converts some of the collagen to gelatin when you're cooking your meat. And tougher meats need more cooking as they have greater cross linkages between these chains. So that's what you're doing when you're cooking your meat, is breaking that down. Immunoglobulins are antibodies produced in our bodies as a response to microorganisms or some foreign molecules that invade our system. The structure of an immunoglobulin is 4 polypeptide chains, 2 identical heavy chains, and 2 identical light chains. So you see here we've got this longer chain here and a longer chain here, and then here are 2 smaller protein chains. And the heavy chains will have around 400 or 500 amino acids, and your shorter chains will be about half that size, so 200 to 250 amino acids in these chains. The main part of these chains will be the same in all the immunoglobulins. It's these ends that differ depending on what antigen you're trying to get rid of in your body. So the antigen is the foreign element that's in your body, and they will bind to these amino ends of these 2 chains in this immunoglobulin. And here's another picture to kind of show you. Here's an antigen kind of bonded in here. Here's another one bonded in there, so forth and so on. So these antigens will bond to these amino sites, and then your body will precipitate these out in order to remove them from your body. I think I skipped over some points here. Constant regions. Yeah, so the sequence here is going to be different depending on the type of antigen that your body is trying to eliminate. Mother's milk includes an immunoglobulin called colostrum, or colostrum has immunoglobulins in it. And this is why nursing is much better for the baby's immune system than just a store-bought formula. So they're getting these immunoglobulins from the colostrum in the mother's milk. Lipoproteins, I had this picture up on the previous slide. These are conjugated proteins because they're made up of lots of different things that have lipids and amino acids in them. And lipoproteins will suspend lipids. This is why they're called lipoproteins. And it will transport those lipids through your bloodstream. And these are often used as an indicator of how high of a risk you are for a heart attack, as they can measure your lipoprotein levels in your system. If it's way too high, then you've got too many lipids in your system, and you're likely to be high in cholesterol, developing plaque in your body. Okay, that's the end of chapter 9. I've got some suggested problems here for you guys to look at for either 4th or 7th edition. It is 8:14. Do you guys want to take a short break before we dive into chapter 10? Yeah. Since you're so quiet, I'll just make an executive decision and say, yeah, why don't we take another 5-minute break? Why don't we come back at 20 after, and we will dive into chapter 10. I know a few weeks ago, my wife said that she was at Meijer, I think it was Meijer, and there was almost no ice cream. there at all. And then a couple days later, I happened to be at a CVS and I looked in the back, and sure enough, their ice cream section was completely empty. So there was like an ice cream shortage going on for a bit. My oldest daughter had her birthday this weekend, so we went and got ice cream. So it's back in stock. But a lot of that going on. Yeah, first day of spring, let's go get ice cream. Yeah, when it was 70 degrees out, I dropped my lid on my Beetle, and we went and got ice cream for sure. Yeah, it's been too cold since then, but it'll warm up. And then it gets too hot and I can't have the lid down. Michigan's not a good place to have a convertible, really. Okay, let's see here. I got resume sharing, resume recording. You guys seen chapter 10, enzymes and vitamins? Okay, so like I said, this is fairly short. There's 28 slides. Enzymes. So enzymes, the word enzyme comes from the Greek enzyme, which literally means in yeast. And yeast is an enzyme that causes bread to rise. It was one of the early enzymes detected. That's why we end up with the word enzyme. Enzymes act as a catalyst for biochemical reactions. Because they are a catalyst, they are not consumed in the reaction. And these reactions in our bodies will happen millions of times faster than they would without the enzymes. So they have a dramatic effect in boosting reaction speed. Each cell in our body has thousands of different enzymes, all for different reactions that need to go on. And they are used with all kinds of reactions, undergo all reactions like proteins, including denaturation. Oh, enzymes will have the same kind of reactions like proteins do, including you can denature enzymes. Like we already said, that's what kills you with a high body temperature is your enzymes no longer being able to function. So enzymes are named according to their function, not their structure. So this is a little different. And usually they get the suffix "ase," but some have the suffix "in," in. So some examples here are oxidase. It catalyzes an oxidation reaction. Hydrolase catalyzes a hydrolysis reaction. Urease does the hydrolysis of urea. Lactase, hydrolysis of lactose. Cellulase, cellulose, sucrase, sucrose, you get the idea. So it's describing the function of the enzyme, not its structure. Aspartate aminotransferase, quite the moniker, is an enzyme that assists in transferring an amino group from aspartate to another molecule. So that's why it's called aspartate aminotransferase. And then we have these different ones that we just looked at. Different classifications of these enzymes. Oxidoreductase will govern oxidation reduction actions. A transferase transfers a functional group from one molecule to another. Hydrolase is used in hydrolysis reactions where water causes bonds to break. A lyase adds a group to a double bond, thereby breaking that double bond, or it removes a group to form a double bond. Isomerase will convert a molecule to an isomer of itself. We're going to see examples of all these. So it just sort of changes the molecule into another, the same molecule but a different isomer. and a ligase bonds two molecules into one, and this usually in our bodies is with the participation of a chemical called ATP, which becomes very important next week. So a simple enzyme is only a protein. A conjugated enzyme has a non-protein part as well. Again, kind of like we talked about conjugated proteins. Conjugated enzyme is just a conjugated protein. The protein part is called the apoenzyme, and the non-protein part is called the cofactor. These cofactors are usually small organic molecules or metal ions like zinc, magnesium, manganese, iron, chloride ions. They provide additional chemical reactive functional groups to the enzyme. And dietary minerals like zinc and magnesium and manganese are an important source of these inorganic cofactors on these enzymes. Being fundamental atoms, your body can't produce these. You have to eat them in order to have them in your system. So a holoenzyme is the whole enzyme. So a holoenzyme is equal to the apoenzyme, which is the protein part, with this cofactor in the case of a conjugated enzyme. The actual active site of the enzyme is a small part of the overall enzyme that is involved. A substrate is the chemical that's undergoing the reaction, and the enzyme will react with that substrate and form a complex momentarily to complete the reaction, and then it will revert back to its natural enzyme shape. This enzyme substrate complex is an intermediate form when the substrate binds to the active site on the enzyme. So here's an example. This is what is called a lock and key enzyme, because only substrates with the same geometry of the enzyme can access the active site. So you can see here, this is just a conceptual shape of a chemical that's going to react. It's kind of got like almost looks like a tooth with a pointy end and a square end. This will come into the active site on an enzyme and see how it's shaped exactly the same way. So this chemical will come in and bind to that enzyme. And then, oh, Lydia, you got your camera working. Sweet. I'm not talking to walls anymore. Thank you, guys. The reaction occurs, and then the products of that reaction pop off again, and the enzyme's back to being the same all over again. So the enzyme forms this complex, the reaction occurs, and then the products come off, and you get the same enzyme that you had before. And this is called lock and key because this is kind of like the lock, and this is like the key. It has to fit exactly. An induced fit enzyme is where the active site morphs to fit the substrate. And this morphing happens through electrostatic interactions, hydrogen bonds, and hydrophobic interactions with the substrate. So you can see here, this now doesn't look, it doesn't have a pointy or a square side yet, but as the chemical comes in, it starts to react with this enzyme, and the enzyme changes shape in order for the attachment to occur. The reaction happens, and then as the products kick off, this goes back to its old shape. And so this is called an induced fit enzyme. where it morphs in order for the reaction to occur. So chemically, this is kind of more what's happening here. We have our substrate. These R groups sticking off of here kind of fit onto that active site, which is a polymer with its own R groups. These yellow R groups are the interactions that bind the substrate to the active site. So you can see here that there's some R chain coming off of this protein here, and there's going to be a bond going on here, 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 and here, and that's the active site where this bonds to. The green R groups, you'll notice, are on the chain but are not participating in this active site or this reaction at all, but they help maintain the three-dimensional structure of the enzyme so that it maintains its chemical reactivity. And then we have these blue R groups are R groups that help determine how soluble the enzyme is. So they're not contributing to the shape, nor are they contributing to the reaction site, but they're helping to determine how soluble this enzyme is in your body. Enzymes have different levels of specificity. So there are absolute enzymes. They only react with one substrate. An example of this is urease. Urease will only react with urea. So this is the urease here, and this is the urea, and it will only act with that. The chemical bonding here is such that it will only interact with urea. A stereochemical enzyme will only react with one stereoisomer. So an example of this is L-amino acid oxidase. So this is involved in the oxidation of L-amino acid. It will not interact with D-amino acid. It will only react with the L stereoisomer. Some enzymes are a little bit broader. They will interact with a group. And so this allows substrates of similar structure to interact. It will interact with more than just one chemical, and it will react with a group of chemicals. An example of this is carboxypeptidase. It cleaves off that carboxy group, that C-terminal end. So this will react with any chemical that has that C-terminal end on it, any protein with a C-terminal end. And then finally, we have linkage enzymes that will work with certain bond types. So for example, phosphatases will hydrolyze any compound that has a phosphate ester bond in it. So these two are much more general. These two are very specific. Any questions there? Okay, factors affecting activity are temperature, pH, the concentration of your substrate that you're wanting to react with, and the concentration of your enzyme. So temperature, you're going to get a bell-shaped curve where the higher the temperature, you will cause the reaction rate to go higher, but you will reach a maximum because then you will start to denature your enzyme and possibly the substrate as well. And so the reaction will quickly fall off at higher temperatures. Does that make sense? So you're kind of warming it up, we're getting good activity, things are colliding more frequently when you increase heat, you hit this optimal temperature, but then you'll quickly start to destroy your reactants, and so you'll lose the reaction. pH, very similar kind of a thing. You're going to have an optimal pH level for the chemicals involved. But again, if you get too basic or too acidic, you're going to cause the reactions to fall away quickly. The substrate concentration, you can add more and more substrate, and initially you'll get a very steep curve because there's usually a lot of excess enzymes in the body, but you will form an asymptotic curve here because there's only so many enzymes. So you will hit this maximum reaction rate when you start to get closer and closer to all the enzymes being used in the reaction. There's no more to react with. Does that make sense? So you'll just end up not going any faster because there's no more enzymes. With enzymes, however, if you increase your enzyme concentration, you will just keep increasing the reaction rate, as assuming you have tons of substrates to react with. And so you get a very linear 45-degree angle here with this. Okay, enzymes can be inhibited. Enzyme inhibitors can slow or stop the catalyst function of an enzyme by bonding to it. There are two types of inhibitors. There's competitive inhibition, and this is when a molecule is similar in shape and charge distribution. So here we have some normal substrate that wants to react at that active site on the enzyme. But this competitive inhibitor is some different chemical, but it has the same structure enough that it will bind to that active site on the enzyme, and therefore this guy can't get in there. Oops. This guy can't get in there in order to react. So it's competing for that active site. A non-competitive inhibitor is when a molecule binds to another part of the enzyme. And so what it can do is another molecule can come in and bind to another place in the enzyme, but that affects this shape. And therefore, this substrate cannot bind into that site, or it's harder for it to bind into that site, and therefore it will slow down or even possibly stop the reaction from happening at all. Any questions there at all? Irreversible inhibition is where a molecule forms a strong bond with the amino acid side chain at the active site, and you can put more and more substrate in, and it's not going to reverse it because it's such a strong bond to it. And like here, some of these guys are going to get into an enzyme in this reaction, but some of them will get to the enzyme first over the competitive inhibitor. So this merely slows down the reaction, and you could speed up the reaction by adding more substrate, because then you're getting more attachments between the substrate and the enzyme, right? But in the case of an irreversible inhibition, it has so affected this enzyme, and it's such a strong reaction that it just completely keeps your substrate from being able to bond at all. And it's such a strong reaction that no matter how much more substrate you add, it's not going to be able to bond with it. Some examples of these are nerve gases and organophosphate insecticides. They will just kill these enzyme reactions. Heavy metals will actually attach to another place. This is arsenic as an example. And it just deforms this active site. But because this is bound so tightly to this enzyme, it's effectively ruined this enzyme forever. This is not going to come off and allow this to go back to its proper shape. Penicillin is an example of this. It binds with a transpeptidase in the bacteria, and that keeps the bacteria from forming its cell walls. So this is how penicillin kills bacteria. Because the bacteria can't form cell walls, then it's very much subject to those walls being broken. They're weaker because of it, and therefore the bacteria just kind of falls apart, and you've rendered it inactive. Aspirin is another one. It will irreversibly bind with cyclooxygenase, and this reduces inflammation. And it's such a strong bonding on that it keeps that inflammation from occurring. Okay, simple enzymes just have an active site. So as we've already seen before, your substrate comes into the active site and has the reaction. You can have a competitive inhibitor that comes in and competes. So we've got the red diamond can compete with this green triangle, and this decreases the rate of reaction. But there are also what are called allosteric enzymes that have an active site and a regulation site. So this regulatory site is another place on the enzyme, and these sites will react with another substrate that will modulate the reaction that happens at the active site. And it can increase or decrease the reaction. So this modulator coming in, attaching here, can make this reaction happen faster or slower. Any questions there? So allosteric enzymes typically happen when you have more than two protein chains. There's a quaternary structure, in other words. There's these two kinds of binding sites on them for substrates and inhibitors. Often one is on each chain. So you'd have your binding site is on one chain and your substrate active site is on the other chain. The binding molecule at the inhibitor site changes that 3D structure, including the active site. So, I mean, we're just drawing these as conceptual blobs, but remember these are proteins with a particular structure. This comes in and that changes the chemistry of this active site to either make it more or less reactive, and it does this by affecting that 3D structure there. Another type of normal inhibition is called feedback control. Quite often reactions happen in the body through multiple steps. And so you have like step A uses enzyme 1 to produce B. This is a different molecule. B then uses enzyme 2 to become a different molecule C. C uses a third enzyme to become molecule D, but D acts as an inhibitor to this AB reaction with enzyme 1. And this is how your body governs so you don't have runaway reactions. Which again, I just find really amazing that your body does this stuff. You know, it's just absolutely incredible. So but what can happen then is this D can get consumed and thereby allowing this reaction to occur again. So it becomes a very much a cycle. So some other process consumes that D, allowing this first reaction to happen again. Any questions there? Okay, zymogens, we're almost done. Zymogens are enzymes that there are proteolytic enzymes that break peptide bonds. So oftentimes you have proteins in your system that start as a zymogen, which is an inactive form of the enzyme. And a piece of it has to be removed in order for it to become chemically active. So this zymogen is a precursor to this proteolytic enzyme, which is your active enzyme. And your body does this, again, very fascinating to me, so that you're not damaging your tissues. If you had a whole bunch of these active enzymes in your body at the wrong time, when your body's not trying to do something that needs that enzyme, then it can do damage to your body. So your body will actually store these enzymes as zymogens, and then when it's needed, it will kick off a reaction to pull a sequence of peptides out of the chain, activating this. Example of this is pepsinogen is a zymogen that gets converted to pepsin in the stomach when it's needed to do digestion. But if it was in your stomach all the time as pepsin, you would end up, it would end up eating your stomach walls. There's not food in there, then it would just start metabolizing your stomach walls. And I looked this up because the last time I taught this class, I said to the class, I wonder if people that have a tendency of producing ulcers have a problem with too much pepsin in their system, you know, like as a genetic abnormality. And I looked it up, and sure enough, that's one of the causes of ulcers, is people can be genetically predispositioned to keeping these in the pepsin state and not the pepsinogen state. And so they form ulcers in their stomach because of it. There's other causes of ulcers too, but this is one of them. Any questions there? Okay. Antibiotics, they kill or inhibit bacterial growth. Some examples of this are sulfa drugs. They inhibit, so bacteria likes to eat para-aminobenzoic acid. This is abbreviated PABA, but this is a favorite food of bacteria. Sulfa drugs, so you see here, this is an example of one, sulfanilamide. It's got this sulfur in here and this structure. This will keep the bacteria from consuming the PABA. And so this is a very effective antibiotic. A lot of antibiotics that are used today are sulfa drugs. And what the bacteria does is it converts the PABA to folic acid. But the sulfa drugs only affect the bacteria because you also eat folic acid. So it's not like taking this sulfa drug is going to put you in a deficit of folic acid because you eat that on a regular basis. And then we talked about penicillin. Penicillin will bond with the wall of a bacteria here, and this inhibits this bacteria from strengthening its cell walls. And therefore, any kind of shock or osmosis will cause what is called lysis, which is breaking open of the cell, and that kills them. So the penicillin comes in and attaches to this active site on the wall of the bacteria. You see it formed here. And then now it can't react with that transpeptide base and strengthen its cell walls. Ciprofloxacin, often abbreviated just Cipro. You can see the structure of it here. Again, I'm not going to test you guys on any of these structures. You know that by now. But ciprofloxacin is very effective in attacking bacteria in the urinary tract, your intestinal system, your intestines, and respiratory systems, your lungs. As a matter of fact, ciprofloxacin is the most common treatment for anthrax, being exposed to anthrax. Anthrax is a respiratory system agent, often used as a biological weapon, and ciprofloxacin will help deal with a patient who's been exposed to anthrax. Medical uses of enzymes. So you can use enzymes to diagnose diseases. Some enzymes found in the blood can indicate where tissue damage is happening and or cells are leaking out. The cells have been damaged themselves. So measuring those enzymes in the body can tell you if that's happening, and therefore they can backtrack to know what disease is causing that. Enzymes can also be used in the treatment of diseases. There is an enzyme called TPA, which activates plasminogen, which is used in your system to dissolve blood clots. So they can treat blood clots by adding this enzyme into your body. And it can be also used in clinical lab analysis. An example of this is urea is converted to ammonia using urease. And what they could do is they could take a blood sample and they can measure the urea in your blood by converting it to ammonia and then measuring the ammonia. It's easier to measure the ammonia than the urea, so they just convert it using a urease, and then they measure the ammonia level in your body, and then they can backtrack it and know how much urea you have in your system. And if you have high levels of urea in your blood, this can indicate that there's a kidney malfunction going on, because your kidneys are used to eliminate urea from your body. Okay, just a few slides on vitamins. Vitamins, oddly enough, the legal definition is an organic compound that must be obtained from your diet. That's all it is. I thought that was kind of weird. Many enzymes have vitamins as part of their structure. Unlike proteins, where you need to eat grams and grams of proteins every day, vitamins you need very little. You can have micrograms or perhaps sometimes milligrams is all you need daily. 1 gram of B12 vitamin could support 500,000 people. So you don't need a lot of B12, but you do need some B12. Contrary to popular myth, synthetic vitamins are just as good as natural ones. They're chemically identical. So you taking your Flintstone vitamin is just as good as eating those vitamins in your food. Doesn't make a difference. There are 13 known vitamins, A, C, D, E, K, and then B1 all the way up to 12, except there's no 8, so forth. These names literally came from the names on the test tubes. So as they were separated out these vitamins and identifying them, it was literally test tube A, B, C, D, and E, so forth and so on. And so vitamin A was just the vitamin that they found in that particular test tube as they were separating them. Vitamin B has numbers because they found out that originally what they thought was vitamin B turned out to be multiple different kinds of vitamins. So you end up with B1, 2, 9, and 12, so on and so on. There are water-soluble vitamins and lipid-soluble vitamins. Water-soluble vitamins must be consumed because your body will excrete them. One of the most famous, well-known ones is vitamin C. Your body cannot store vitamin C, so you have to be taking in vitamin C on a regular basis. And vitamin C is important for the formation of skin, ligaments, tendons, bone, and teeth. So these are like these fibrous proteins. It also acts as a general antioxidant. So vitamin C will oxidize before other chemicals, thereby protecting them. Vitamin C is also involved in the metabolism of several amino acids, converting them to hormones. Vitamin C, by the way, is the lack of vitamin C. What do you develop if you don't have vitamin C? What disease do you get ultimately? Sailors used to get it.
Scurvy.
Scurvy, exactly right. The sad story about this is there was a British naval doctor that figured out what was causing scurvy, and he recommended it to the Admiralty, and he was laughed at so much it destroyed his career. And he ended up dying with the broken man. 30 years later after his death, they realized he was right all along. And they started giving sailors a spoonful of like lemon juice or orange juice every day, and scurvy went away on Navy ships completely. Because again, you have to ingest vitamin C on a regular basis. Your body can't store it. Vitamins B2, 3, 5, 6, 7, 9, and 12. They have to be chemically altered before they're useful within your body. They are used to transfer elements or functional groups throughout your body. And B3 and 9 are particularly important for cardiovascular health. Fat-soluble vitamins are A, D, E, and K. They get stored, these can be stored in your body, and they're stored in the fat tissues of your body. Vitamin A is very good for vision. It's specialized immature cells. Oh, it's used for immature cells in your body to be, cells can be have like an immature state, and then they can turn into different types of cells. And vitamin A is used in specializing these immature cells into different kinds of cells that your body needs. It's also used in the formation of skin, linings of your stomach and lungs. Vitamin D is made mostly within the body with UV light. This is why you, in the wintertime, a lot of people take vitamin D because you're not exposed to as much UV radiation as you are in the summer. And vitamin D is used in your body to maintain your calcium and phosphorus ion levels in the blood so that your bones can absorb those nutrients. So I wonder if that means that that's like I think it probably is. Don't older women usually take vitamin D because of osteoporosis? So I believe that's true. I'm trying to remember if my wife takes a vitamin D. That could be to help, the body continue to build stronger bones. Vitamin E acts as an antioxidant in the body. Vitamin K contributes to blood clotting, the synthesis of proteins in blood plasma, bone, and the formation of kidneys. And we are at the end. Here is some problems for you guys to take a look at, depending on which edition you're using. So again, the exam will kick off Friday, go all weekend, take it anytime. The exam's over chapter 7 and 8, not stuff we covered tonight. And next week we will cover chapter 11 and review some of this stuff as well. Any last comments, questions, anything? If not, we're done.
Thank you.
Thank you.
Yep. Have a good night,
you.
everyone.
Night.